This is a video for Science 9, and we're looking at what happens during cell division. So we've got our cell here with the DNA and a nuclear membrane, and here's our outer boundaries of our cell. So what has to happen first is it has to be big enough, it has to have enough energy, and there has to be a need for a new cell. So the first thing that will happen is our cell grows, and then we have to make a copy of all of our DNA. So our DNA is represented as all of this wool, so we're going to throw in another copy of each of our strands of DNA. And now our cell has replicated its DNA. And often in biology we ask ourselves, well, what if it wasn't like that? So usually we see in the books that it condenses into chromosomes, but what if it didn't do that? So we have to think about our cell and, how could it equally divide all of this DNA without getting tangled? If we didn't wrap it up into chromosomes, we would get knots, we would get breakage, and we definitely wouldn't get things in an orderly way to move to different parts of the cell. So after all of the replication has happened, the cell enzymes have checked for mistakes and made any corrections, we have to bundle up our DNA in an orderly way. If we didn't bundle it up, we would end up, if you've ever had a snarl of yarn, you're going to get some tangles. It would be really difficult to get everything divided up equally and move to a new cell. It moved to the other side. So we're going to have to condense our DNA into chromosomes, kind of like this. So we have our replicated chromosomes here. And the next thing we need to do is line them up along the middle so that they can be separated evenly. So we have to, first of all, dissolve our nuclear membrane, because that can't be there. And then we're going to line up our chromosomes along the equator of our cell. So how do they get to the other side? Well, we have these spindle fibers that attach to the centromeres. That would be the elastics on our wool bundle. So these have formed, and eventually they're going to shorten and our DNA will migrate to opposite ends of the cell. Now, we can't leave things like that. When our DNA is bundled up like that, we can't have any protein synthesis. So we need to form new nuclear membranes around our DNA. Our DNA has to unwind itself. And finally, our new cells need to partition each other. So we're going to start to grow a new set of membranes that will create our new cells. Because think about it, what if it didn't do this last step? What if the nucleus kept dividing and dividing? Well, eventually our cell would get too big and it would have too many nuclei who would be in charge. So anyway, here is how we get our two duplicated cells. And if you have a cell wall, there's going to be cell wall growing in between here. And you have your two identical daughter cells.